For more than 500 years, sailors have crossed a region of the North Atlantic that behaves unlike any other sea on Earth. In 1492, Columbus and his crew entered it and found the surface covered in drifting brown gold weed, with no coastline in sight and vegetation stretching toward the horizon. Some aboard feared they were approaching land or becoming trapped far from it. But the history of this place extends far beyond the sailors who eventually gave it a name. For millions of years, four enormous currents have been circling this part of the Atlantic, enclosing a body of water without building a single shore around it. The Gulf Stream moves along one side, the North Atlantic Current crosses another. The Canary Current descends along the east, while the North Atlantic Equatorial Current completes the enormous clockwise circulation. Inside them sits the Sargasso Sea, the only named sea on Earth whose boundaries are made entirely of moving water. At the surface, floating sargassum forms a golden forest across immense stretches of open ocean. Beneath it, the water descends through clear blue, then twilight, then complete darkness, eventually reaching depths approaching 15,000 feet. And what scientists have discovered between those two extremes has made this sea considerably stranger than the old stories surrounding it. There is an animal hidden inside the floating weed that has been found consuming its own young. There is a biological mystery connected to these waters that Aristotle was already writing about more than 2,000 years ago, and parts of it remain unresolved. And when scientists began examining what appeared to be ordinary Sargasso seawater at the genetic level, they uncovered an invisible world whose diversity was far beyond what they expected to find. The deeper science looks into the Sargasso Sea, the less ordinary it becomes. Tonight, we are descending through that hidden world, from a floating forest exposed to sunlight to an abyss where much of the life below has rarely, if ever, been observed directly. But before we continue, let me know in the comments if you have any topic suggestions you would like me to cover in future videos. Let's descend into the currents themselves, because before we can understand what the Sargasso Sea is hiding, we need to understand how a sea with no coastline can exist at all. A sea does not always need a shore to give it shape. In the central North Atlantic, moving water performs the work that continents usually do. Currents curve around an immense region of open ocean, forming boundaries that shift and breathe rather than remaining fixed against rock. Within their circulation lies the Sargasso Sea, a body of water whose geography is written almost entirely in motion. Its broad domain extends roughly 700 miles from east to west and nearly 2,000 miles from north to south, although its edges wander with the currents. It occupies the interior of the North Atlantic subtropical gyre, generally between about 20 and 35 degrees north latitude and 40 to 70 degrees west longitude. Those coordinates describe an enormous expanse, but they do not mark a wall. Crossing into the Sargasso can happen without any visible sign that one ocean region has ended and another has begun. Four great current systems shape this watery enclosure. Along the west, the Gulf Stream carries warm water northward with remarkable force. Farther north, the North Atlantic Current continues the circulation eastward. The Canary Current turns cooler water south along the eastern side, while the North Equatorial Current helps complete the broad clockwise circuit toward the west. Together, their movement forms the gyre, a rotating ocean system whose interior behaves differently from its energetic margins. Rotation gradually draws surface water toward the gyre's interior. Winds and Earth's rotation help produce this convergence, causing water to accumulate slightly toward the center and then sink. The result is not a motionless pool, but a vast region where horizontal circulation is comparatively sluggish and where material drifting at the surface can become concentrated. The sea therefore has boundaries without beaches, maintained by physics rather than stone. Bermuda rises near the western portion of this open water realm, the conspicuous patch of land associated with an otherwise pelagic landscape. Beyond it, there is no continental margin enclosing the Sargasso. Its identity comes from oceanography itself, making it unusual among the world's named seas. This circulation also helps create the character of the water. Nutrients that support abundant surface growth are scarce through much of the central gyre. Strong layering separates warmer upper water from colder water below, limiting the upward delivery of nutrients from depth. With relatively little suspended biological material, sunlight penetrates unusually clear blue water. The apparent purity is partly a sign of scarcity, an open ocean environment where life must make efficient use of what little nourishment circulates near the surface. Floating within this nutrient-poor expanse is sargassum, the brown algae that gave the sea its name. Portuguese mariners associated its small gas-filled bladders with grapes, and the name endured. Unlike seaweeds anchored to coastal rock, the dominant floating forms here spend their lives adrift. Their buoyant structures hold branching fronds near sunlight, and fragments can continue growing vegetatively without requiring a seabed. That floating existence suits a region built from circulation. Water, organisms, drifting plant matter, and debris can linger within the broader gyre for long periods, although residence times vary greatly with depth, location, and the constantly changing currents. Nothing here is sealed completely. Eddies exchange water across the margins, storms disturb the upper layers, and currents meander from year to year. Yet the persistent clockwise circulation repeatedly recreates the same broad oceanographic province. Its foundations are ancient. The modern current system developed through a long history of continental movement, changing gateways, winds, and climate after the Atlantic opened many tens of millions of years ago. The Sargasso of today is therefore not a preserved fragment unchanged since deep time. It is a living arrangement of water, continually rebuilt by forces acting across an ocean basin, a sea whose borders exist because the Atlantic never stops moving. From the deck of a ship, sargassum can seem almost insubstantial, a scattering of amber fragments riding the Atlantic swell. Seen at the scale of the animals that depend upon it, the same vegetation becomes architecture. Branches divide into corridors, overlapping fronds form ceilings, and round gas bladders hold the entire structure near sunlight. A habitat only inches thick can contain more biological complexity than the open water surrounding it for miles. The floating growth belongs mainly to two forms, Sargassum natans and Sargassum flutans. Unlike most familiar seaweeds, these forms spend their lives without attachment to the sea floor. They remain buoyant through small gas-filled structures called pneumatocysts, which lift their leaf-like blades toward the sun. Broken pieces can continue growing vegetatively, allowing drifting fragments to become living habitat rather than dying debris. Their brown and golden appearance comes partly from pigments adapted to capturing light. Chlorophyll remains present, but other pigments, particularly fucoxanthin, mask much of its green color. Through photosynthesis, the algae transforms sunlight and carbon dioxide into organic matter, adding both food and structure to an ocean region where concentrated habitat is scarce. This was the material encountered by Christopher Columbus and his crew in 1492. To sailors crossing an unfamiliar Atlantic, broad accumulations of vegetation suggested that land might be nearby, yet the plants required no nearby coast. Their presence marked an ecosystem suspended entirely at the surface, carried wherever winds, waves, and circulation rearranged it. Individual pieces often gather into elongated rows called windrows. Waves, surface currents, and wind can push scattered plants together until loose fragments become larger aggregations. Within those tangles, the physical world changes. Sunlight becomes broken by overlapping blades. Tiny pockets provide shelter from larger hunters. Surfaces become available for animals to cling to, graze upon, hide beneath, or use as nurseries. A clump no larger than a soccer ball can host an extraordinary congregation. Small crustaceans move among the branches. Snails graze across algae and attached organisms. Worms, bryozoans, hydroids, shrimp, crabs, juvenile fish, and other invertebrates occupy spaces that would otherwise be featureless water. Some residents remain associated with sargassum for much of their lives, while others use it only during vulnerable juvenile stages. Fish diversity is especially rich around these floating structures. Surveys have recorded well over 100 species associated with sargassum communities, including juveniles of economically important and highly mobile fishes. Young dolphinfish, jacks, filefish, triggerfish, and other species can find both prey and concealment among the vegetation. Juvenile sea turtles also enter these drifting habitats, where the floating material offers food and a three-dimensional refuge unavailable in surrounding open water. The community survives partly through intense recycling. Nutrients released by animals become available to microorganisms and algae. Epiphytic organisms grow directly upon sargassum surfaces. Small grazers consume this growth and organic particles, then become prey themselves. Material that might disperse through open water is repeatedly captured, consumed, excreted, decomposed, and used again within a compact biological neighborhood. That efficiency matters because the surrounding ocean contains comparatively little nutrient supply near the surface. The contrast is striking. Sparse open water can lie immediately beside a floating concentration of shelter, food, eggs, larvae, grazers, and predators. Marine biologist Sylvia Earle has described sargassum habitat as a golden floating rainforest, an image that reflects its role more than its appearance. Yet this forest has no trunks and no soil. Its pathways separate and reconnect with every passing swell. Entire neighborhoods can fragment overnight, then gather elsewhere under changing winds. For the organisms living among its branches, home is not a location fixed on any map. It is a drifting structure of living brown algae, carrying a concentrated community across an otherwise immense expanse of blue. At the scale of a small crustacean, a strand of sargassum is not vegetation drifting through water, it is terrain. Every fork in the algae creates an intersection. Every curled blade becomes cover, and every cluster of gas bladders breaks the outline of whatever waits beside it. Survival here depends less on speed than on becoming difficult to distinguish from the habitat itself. Natural selection has refined that disguise across many unrelated animals. Browns, yellows, creams, and irregular patches repeat the colors of aging fronds. Some bodies are translucent. Others carry knobs, filaments, or ragged edges that interrupt the familiar outline of a fish or shrimp. Even movement can betray an animal. So many residents creep with the swaying vegetation rather than swimming openly through the spaces between branches. Few demonstrate this better than the sargassum fish, Histrio histrio. It belongs to the frogfish family, yet its body seems assembled from pieces of the weed around it. Mottled skin breaks into irregular markings, while fleshy projections resemble surrounding growth. Instead of relying entirely on swimming, it can grip and clamber through the algae with modified pectoral fins, placing itself among the branches with remarkable control. Once positioned, the fish can remain almost indistinguishable from its surroundings until prey comes within reach. Like other frogfishes, it possesses a modified dorsal spine that functions as a lure. A small appendage at its tip can be moved near the mouth, drawing the attention of animals that mistake the motion for something edible. The camouflage that protects the predator therefore becomes part of the hunting mechanism itself. The Sargassum pipefish follows another strategy. Its narrow body resembles the elongated pieces of vegetation around it, and its restrained movements preserve that resemblance. Rather than chasing prey, the pipefish approaches tiny crustaceans with a tubular snout. Rapid expansion inside the mouth creates suction, pulling the target inward before a prolonged pursuit is necessary. Marine biologist Wolfgang Sterer compared the action to a powerful pipette, an apt description for feeding performed with such concentrated precision. Shrimp achieve concealment with less elaborate anatomy. The Sargassum shrimp, Leander tenuicornis, carries much of its camouflage within nearly transparent tissue. Yellowish and brown markings disrupt what remains visible, allowing the animal to merge with the colors beneath and around it. Against a moving background of translucent water, shadow, and algae, transparency can be more useful than a solid imitation. Not every adaptation within the mat belongs to a hunter. Flying fish can attach masses of eggs to floating material, including sargassum. Fine adhesive filaments secure the eggs to the vegetation, preventing them from drifting individually through exposed water. The nursery is therefore woven directly into a habitat already occupied by grazers, scavengers, predators, and other developing animals. That arrangement creates an unusual intimacy between concealment and danger. The same branch that hides a juvenile fish can conceal the mouth waiting for it. A shrimp's protective transparency can help it approach smaller prey while reducing its visibility to larger hunters. Camouflage has no allegiance within the mat. It favors whichever animal uses it successfully for another moment. Some members of this community are so closely associated with pelagic sargassum that their appearance, behavior, and life histories make little sense away from it. Generations have unfolded on vegetation that never roots, producing specialists adapted not to reef, sand, or coast, but to a habitat whose branches are always traveling. Within that shifting structure, resemblance becomes a form of currency. A fraction of an inch in the wrong direction can expose a body. Remaining alive may depend on matching a brown blade, moving when the algae moves, and allowing another animal to pass close enough without recognizing what it has seen. Cannibalism leaves a peculiar record in preserved specimens. Long after the moment of feeding has passed, the stomach can retain evidence of an encounter that no observer witnessed. For scientists studying frogfishes associated with floating sargassum, those contents have revealed how far predation can extend when nearly anything small enough to swallow can become food. The animal most closely specialized for life among pelagic sargassum is the sargassum fish, histrio, a small member of the frogfish family. Its resemblance to the surrounding algae is more than protection. It allows the fish to remain concealed while prey moves within striking distance, turning the vegetation itself into part of an ambush. A frogfish does not need a prolonged chase. When suitable prey approaches, its mouth expands with extraordinary speed. The jaws open, the oral cavity enlarges, and water rushes inward with the victim. In some frogfishes, the feeding sequence unfolds within only a few thousandths of a second. Rather than seizing prey between conventional biting jaws, the fish creates suction powerful enough to draw an animal directly into an expanding mouth. That feeding mechanism is paired with an unusually accommodating body. Frogfishes can consume prey remarkably large relative to themselves. Their stomachs expand, allowing a meal that would appear disproportionate beside the predator before ingestion. Fish, shrimp, and other inhabitants of the floating community can disappear into a hunter whose outline had been almost impossible to separate from the algae moments earlier. The same capacity creates another possibility. A smaller member of its own kind still possesses flesh, protein, and energy. Evolution does not require a predator to recognize kin as exempt from predation unless preserving that relative provides a greater reproductive advantage. Cannibalism is well documented among frogfishes. Larger individuals may swallow smaller ones, and reports from natural history observations include striking cases in which numerous young were recovered from the digestive tract of an adult. Such findings are sometimes described as parental cannibalism, although interpreting the exact relationship between predator and juveniles requires care. A stomach can preserve what was eaten without preserving the circumstances that brought the animals together. For the sargassum fish, reproduction itself creates moments when adults and offspring can occupy the same drifting environment. Females release eggs within a gelatinous ribbon or raft that floats near the surface. The eggs develop within this temporary structure before the young emerge into pelagic life. There is no prolonged parental care protecting each juvenile after hatching. Once independent, a small fish enters the same ecological network of prey and predator as everything around it. Cannibalism can provide several evolutionary advantages in fishes. It can recover energy when alternative prey is limited, reduce competition, and allow a larger individual to exploit a readily available food source. None of this requires planning or cruelty. Natural selection acts through consequences. Individuals whose feeding behavior helps them survive and reproduce can pass those tendencies onward, even when the meal happens to share their ancestry. There are costs as well. Consuming relatives can reduce an individual's indirect genetic contribution to future generations, and eating members of the same species may increase exposure to parasites or disease. Cannibalism therefore persists where its immediate benefits can outweigh those losses under particular ecological conditions. Inside sargassum, those conditions can change from hour to hour. A mat may gather prey, then separate under waves. Small animals can arrive with drifting vegetation or vanish when fragments divide. Food abundance is uneven, creating brief concentrations followed by leaner intervals. For a frogfish wading among the branches, hunger does not recognize the categories humans place upon a meal. Shrimp, juvenile fish, and sometimes another frogfish enter the same field of opportunity. Beneath the mottled skin and elaborate disguise is a digestive system responding to one ancient calculation: whether enough energy can be taken from the life passing within reach. A loggerhead hatchling enters the Atlantic weighing little more than an ounce. Its shell is small enough to rest in a human palm, yet within hours of leaving the beach, it must begin a journey governed by waves, currents, temperature, and instinct. For young loggerheads from nesting grounds along the western North Atlantic, that first journey often leads toward floating sargassum and the circulating waters offshore. The passage begins with frantic swimming. Newly emerged turtles cross the beach, reach the surf, and continue outward through breaking waves. During this early swimming frenzy, they use wave direction and Earth's magnetic field as orientation cues. Once beyond coastal waters, their relationship with the ocean changes. Swimming still matters, but broad currents become powerful transportation systems, carrying the hatchlings toward regions where drifting vegetation offers something the exposed surface cannot. For much of the 20th century, scientists knew surprisingly little about what happened after young turtles disappeared offshore. The interval became known as the lost years, a name reflecting a gap in human observation rather than an absence from the ecosystem. Juveniles could remain beyond easy reach of researchers for years, growing from palm-sized hatchlings into animals large enough to return toward coastal feeding grounds. Research gradually placed sargassum near the center of that hidden period for many North Atlantic turtles. Young loggerheads actively associate with floating weed, where small prey can be found among the fronds. The vegetation also breaks up their outline from animals approaching through the water and provides structure where little exists otherwise. Hawksbill and green turtle juveniles have also been recorded using floating sargassum, although their patterns of habitat use differ. Temperature adds another dimension. Small turtles cannot regulate body heat internally as mammals do, so the thermal character of their surroundings affects metabolism and growth. Researchers associated with the Archie Carr Center for Sea Turtle Research at the University of Florida have examined how young turtles use sargassum habitat and how conditions around these surface communities influence early development. Floating vegetation can create a distinctive microhabitat shaped by sunlight, water movement, food, and cover. Shelter, however, is never absolute. A young turtle entering vegetation also enters a concentrated biological community. Predatory fishes patrol beneath floating material, while smaller hunters occupy the weed itself. Birds can attack from above. Larger fish can approach from below. The structure that reduces exposure in one direction can place a hatchling near organisms searching the same habitat for food. Survival, therefore, combines concealment, movement, feeding, and growth. Body size changes vulnerability. A hatchling that becomes larger gradually escapes some predators capable of taking it during its smallest stages. Food gathered around floating habitat supplies energy for that transition, while warm surface conditions influence how rapidly that energy can be processed. The journey is not simply a passive ride. Experiments have shown that juvenile sea turtles respond to magnetic signatures associated with different parts of the North Atlantic gyre. In-laboratory magnetic fields, representing locations along their migratory route, hatchlings alter swimming direction in ways that would help keep them within favorable circulation. Their inherited magnetic map appears to provide regional instructions, directional responses capable of reducing the chance that a tiny traveler will be swept into dangerously cold or otherwise unfavorable waters. Eventually, growing juveniles begin shifting toward coastal habitats. The timing varies among individuals and populations, and modern research has replaced the older image of turtles spending their early years merely drifting. They swim, orient, select habitat, and respond continuously to an ocean encountered without parental guidance. For the smallest among them, sargassum is neither sanctuary nor trap. It offers food and concealment while gathering competitors and predators into the same narrow realm. A hatchling survives there because evolution has equipped a remarkably small animal to read currents, temperature, magnetic fields, and opportunity within a world that never remains in one place. Remove the floating vegetation from view, and the character of the Sargasso changes almost immediately. Beneath the surface habitat lies water of unusual optical purity, a blue produced not by abundance, but by absence. Suspended sediment is scarce. Large plankton blooms are uncommon across much of the central gyre. Sunlight, therefore, travels farther before particles scatter or absorb it, leaving an expanse that can appear almost empty. Visibility can approach 200 feet under favorable conditions. Red wavelengths disappear first as sunlight enters the water, followed progressively by orange and yellow. Blue penetrates farther, so the remaining light becomes increasingly monochromatic with depth. Objects lose warm colors long before their outlines disappear. A red animal may become dark gray, while pale surfaces acquire the blue cast of the surrounding water. This clarity reflects an ecosystem constrained by chemistry. Near the surface, microscopic phytoplankton require sunlight, nitrogen, phosphorus, and other nutrients to grow. Sunlight is abundant, but many essential nutrients are not. They accumulate deeper in the ocean as sinking organic material is consumed and decomposed, while the illuminated upper layer remains comparatively depleted. Between these regions lies a strong thermocline, a depth range where temperature falls rapidly. Warm surface water is less dense than colder water beneath it, and that density difference resists vertical mixing. Instead of deep nutrients being continually returned towards sunlight, much of their supply remains separated from the organisms that could use them near the surface. Productive coastal seas often behave differently. Winds can drive surface water away from shore and allow colder, nutrient-rich water to rise in its place. Phytoplankton respond rapidly, sometimes multiplying until their abundance alters the color of the sea. In the central Sargasso, persistent stratification suppresses much of that upward exchange. Biological production continues, but it is supported by exceptionally efficient nutrient recycling and by tiny organisms adapted to living with very little. The transition downward is gradual to human eyes. Around 650 feet, enough sunlight may remain for vision, but illumination has weakened substantially. Descending towards 1600 feet brings the water into a twilight realm where daylight becomes faint and increasingly blue. Near 3,300 feet, sunlight useful to vision has effectively vanished. Temperature changes with the light. Surface conditions vary with season and location, while deeper water approaches the cold stability characteristic of the interior Atlantic. The thermocline is therefore more than a temperature boundary. It divides two biological economies, one powered directly by sunlight and another increasingly dependent upon material produced above. Every particle sinking through that boundary carries potential energy downward. Dead plankton, fecal pellets, fragments of tissue, mucus, and other organic remains form what oceanographers call marine snow. Much of it is consumed or decomposed before reaching great depth. Bacteria process dissolved material. Zooplankton intercept particles. Larger animals consume those grazers. Carbon and nutrients are repeatedly transformed as gravity draws the remaining material farther from the illuminated surface. Scarcity shapes body size, metabolism, feeding behavior, and movement throughout this column. An animal living where meals arrive unpredictably cannot spend energy as though food were constant. Some reduce activity. Others travel vertically to exploit richer layers during particular hours. Many depend indirectly on production occurring hundreds or thousands of feet above them. The surrounding gyre influences this biological structure, but the deep Sargasso is not completely sealed from the Atlantic. Water masses circulate beneath the surface system, carrying oxygen, heat, dissolved chemicals, and organisms across enormous distances. Eddies also disturb boundaries and occasionally inject nutrients into upper waters, creating temporary pockets of greater productivity. So, the emptiness is never absolute. The blue water is better understood as a landscape of extreme dilution, where life is spread thinly across tremendous volume. Near the surface, sunlight exceeds the nutrients available to exploit it. Farther down, nutrients become more abundant while light disappears. Between those opposing limits, organisms occupy narrow opportunities, following food and energy through a water column whose apparent clarity conceals a finely layered ecology. Dusk changes the ocean long before darkness reaches its deepest layers. As illumination weakens above the Sargasso Sea, countless small bodies begin moving upward through the water column. They do not travel together as a visible procession. They are scattered across immense volumes of ocean, yet their movements follow the same daily rhythm, producing one of the largest coordinated migrations of animal life on Earth. Much of this activity begins within the mesopelagic zone, extending from roughly 650 feet to about 3,300 feet below the surface. During daylight, many inhabitants remain in its deeper reaches, where diminished illumination makes them harder for visual predators to detect. As evening arrives, they rise toward shallower water, following food upward while accepting the risks that come with leaving deeper refuge. Lanternfish are among the most important travelers. Members of the family Myctophidae occur throughout the world's oceans and represent an enormous share of deep-water fish abundance. Most are small enough to fit easily in a human hand. Along their bodies are light-producing organs called photophores, arranged in patterns that vary among species and may assist with camouflage, recognition, and communication. They rise alongside bristlemouths, small squid, copepods, amphipods, shrimps, and other drifting or swimming animals. Some travel hundreds or even thousands of feet between afternoon and night. For a creature only a few inches long, the vertical distance can represent many thousands of body lengths, repeated once upward and once downward during every daily cycle. Their timing follows light rather than a clock. Cloud cover, moonlight, latitude, season, and water clarity can influence when particular species begin moving. Many start ascending near sunset and remain in shallower layers through the night, feeding where microscopic plants and grazing zooplankton are more available. Before daylight strengthens, the migration reverses. Animals descend again, leaving upper waters increasingly sparse. This behavior is known as diel vertical migration, from a word referring to the daily cycle. Its scale is difficult to observe directly because so much of the movement occurs after sunset and beneath the surface. Early oceanographers discovered evidence of it through sonar. Instruments detected a dense acoustic layer that appeared to rise during evening and sink after sunrise. At first, researchers sometimes mistook these echoes for the seafloor. The apparent bottom was alive. The biological purpose is a balance between feeding and concealment. Remaining near productive surface waters continuously would provide greater access to food, but daylight would also expose small animals to predators. Staying deep throughout the night would reduce that exposure while sacrificing feeding opportunities. Vertical migration divides those pressures across different hours. In the nutrient-poor Sargasso, this movement performs another service. An animal may feed near the surface at night, then carry that newly acquired organic carbon hundreds of feet downward before digesting, excreting, or becoming prey. Carbon that entered living tissue through photosynthesis can therefore move into deeper water without waiting for a dead organism to sink. Respiration at depth releases some of that carbon as carbon dioxide. Waste becomes food for microbes and other animals. Predators intercept migrants and incorporate their tissues into deeper food webs. Through countless individual journeys, the migration acts as a biological pump, connecting layers that would otherwise exchange energy more slowly. The magnitude extends far beyond the Sargasso. Estimates of migrating biomass vary substantially because the deep ocean remains difficult to measure, but the movement involves immense numbers of animals across the global ocean each day. No herd crosses a plain, and no flock marks the sky. Much of the migration consists of creatures only inches long. By sunrise, many have returned hundreds of feet downward, Their nightly feeding has redistributed energy, nutrients, and carbon through the water. Above them, daylight resumes its familiar pattern. Below, an enormous living population settles into deeper water, conserving energy until fading sunlight once again changes the balance between hunger and visibility. At roughly 3,300 feet beneath the Sargasso Sea, daylight ceases to be part of biology. From this depth downward, an animal can live its entire existence without seeing the sunday. The water remains near 39 degrees Fahrenheit, and pressure has climbed to roughly 100 times that experienced at the surface. Here, evolution has worked without landscapes, seasons of light, or plants, shaping bodies around scarcity and darkness. Oceanographers call this the bathypelagic zone. Photosynthesis is impossible, so nearly every usable calorie ultimately arrives from elsewhere. Organic particles descend from upper waters, migrating animals carry energy downward, and occasional carcasses deliver brief concentrations of food. Between such opportunities, meals can be separated by long intervals. A successful predator cannot assume that another suitable animal will soon pass within reach. This uncertainty is written into anatomy. Many bathypelagic fishes possess large mouths and expandable stomachs, allowing them to exploit prey that seems disproportionate to their own size. Their muscles are often less substantial than those of fast surface hunters because prolonged pursuit would consume precious energy. Instead, many deep predators wait, drift, sense movement, and commit when an opportunity enters range. The gulper eel, Uropharynx peleconoides, carries this principle to an extraordinary form. Its enormous jaws are attached to a loosely constructed head, producing a mouth far wider than the narrow body behind it. The lower jaw resembles a flexible pouch, while the stomach can stretch after a substantial meal. Its tail extends into a long filament and ends in a light-producing organ that may help attract prey. Much of the animal appears designed around encountering food rarely and making the encounter count. The fangtooth, Anoplogaster cornuta, solves scarcity differently. Adults possess immense teeth relative to their body length, among the largest proportional teeth known in marine fishes. The lower fangs are so elongated that specialized cavities in the roof of the mouth accommodate them when the jaws close. Its compact body and formidable dentition suit a predator that cannot afford an easy meal slipping free. Eyes reveal another set of compromises. Complete blindness is advantageous in some deep animals, particularly where other senses can replace vision, but many bathypelagic fishes retain functional eyes because biological light still matters. Some species enlarge them to gather scarce photons. Others possess tubular eyes aimed toward likely sources of prey. Vision has not disappeared from the midnight zone. It has been redesigned for flashes rather than sunlight. Among the stranger inhabitants is Vampiroteuthis infernalis, the vampire squid from hell. The name suggests a conventional squid, yet the animal belongs to its own surviving lineage, Vampiromorpha, distinct from modern octopuses and true squids. Its 8 arms are joined by a web of skin, and mature individuals possess a pair of retractile sensory filaments, unlike the feeding tentacles of ordinary squid. Its diet is equally unexpected. Rather than behaving primarily as an aggressive hunter, the vampire squid collects drifting organic debris called marine detritus. Sticky mucus combines this material with particles gathered by its filaments, producing food from scraps descending through the water. When threatened, it can turn its webbed arms outward around the body, exposing rows of fleshy projections while protecting the head. Such forms can appear distorted only because human expectations were shaped at the surface. In the bathypelagic world, Enormous jaws, reduced musculature, expandable tissues, sensitive eyes, and unfamiliar feeding structures are not deviations from a normal design. They are solutions to an environment where pressure is immense, food is intermittent, and solar energy never arrives directly. The midnight zone, therefore, has its own logic. Bodies become economical where energy is costly, flexible where opportunity is unpredictable, and exquisitely sensitive where information is scarce. What seems unrecognizable from above is simply life fitted closely to the conditions surrounding it. At the deepest reaches beneath the Sargasso, the ocean finally meets the floor. Nearly 15,000 feet of water may stand above the sediment, placing every square inch under pressures approaching 6,600 pounds. Temperatures remain only a few degrees above freezing. There is no seasonal sunrise here, no storm passing overhead, and almost no trace of the surface except what eventually falls from it. The landscape is called an abyssal plain, although plain does not mean featureless. Fine sediment settles across broad stretches of seafloor, interrupted by rocky exposures, slopes, channels, and isolated hard surfaces where attached organisms can grow. Much of that sediment originates as microscopic mineral grains, skeletal remains, and organic particles descending through the ocean over years. Accumulation can be extraordinarily slow, preserving a record of events far above. Food reaching this depth has already passed through layers of consumers. By the time organic matter arrives, much of its usable energy has been removed. Bacteria and archaea continue breaking down what remains, while worms, crustaceans, echinoderms, and other bottom dwellers search sediment for edible material. Some microbes obtain energy through chemical reactions involving compounds in seawater or seabed material, allowing biological activity to continue without direct dependence on sunlight at the point of consumption. Large falls create temporary abundance. The body of a whale reaching the seafloor can support scavengers for years, then nourish specialized communities as fats trapped within bone are processed by microbes. Smaller carcasses provide briefer feasts. In a habitat where ordinary food arrives as scattered particles, a concentrated mass of tissue becomes an ecological event. Direct observation remains rare. Research vessels can map broad topography with sonar, but identifying animals requires cameras, remotely operated vehicles, autonomous instruments, trawls, cores, or other sampling methods. Each method reveals only part of the landscape. A camera sees a narrow corridor. A trawl samples selectively and can damage delicate organisms. A sediment core captures an area scarcely wider than the instrument itself. Long-term research near Bermuda has provided exceptional knowledge of the water column, yet the biological geography of the surrounding abyss remains fragmentary. Repeated sampling along established routes gives scientists valuable comparisons through time, but repetition also illustrates the scale problem. A narrow transect can be studied carefully while enormous neighboring areas remain biologically unexplored. Deep expeditions elsewhere across the North Atlantic show what such gaps may contain. Glass sponges anchor delicate mineral skeletons to hard substrate. Deep water corals build slow-growing structures without sunlight. Dumbo octopuses, members of the genus Grimpoteuthis and related groups, move with rounded fins above the bottom, conserving energy through gentle propulsion. Eels and other fishes pass through camera frames that may represent only seconds of observation in lives spent across vast terrain. Even tiny drifting animals add unexpected signals. The copepod Gausia princeps, collected from deep Sargasso waters during marine zooplankton surveys, can produce bioluminescent secretions. Light at these depths is biological rather than solar, generated for defense, communication, attraction, or other functions that researchers are still disentangling across species. A long study of deep animals off California found bioluminescent capability widespread among observed organisms, but applying an exact percentage from one ocean region to the Sargasso would go beyond the evidence. The broader lesson remains secure. Biological light is common in the deep sea, and darkness to human eyes does not mean an absence of visual signals. What is missing from scientific records is measured not only in species names, but in untouched space. A vehicle can cross a strip of bottom and document everything its lamps reveal, while terrain beyond that narrow illumination remains unsampled. Beneath miles of Sargasso water, the map may show depth and contour with increasing precision, yet the living inventory remains sparse, assembled encounter by encounter from an ocean floor, far larger than the paths humans have examined. A flash lasting less than a heartbeat can carry more information in the deep ocean than an entire landscape illuminated by day. Far beneath the Sargasso, where sunlight no longer participates in life, organisms manufacture their own signals. A pulse can reveal prey, confuse a pursuer, identify a mate, or erase the outline of a body drifting overhead. This ability is bioluminescence, light produced by chemical reactions inside living organisms or by luminous microbes living with them. Many systems involve a light-producing molecule called luciferin, sometimes working with an enzyme called luciferase, although the chemistry differs greatly among lineages. Unlike an incandescent flame, these reactions release remarkably little energy as heat. Most marine bioluminescence appears blue or blue-green, wavelengths that travel efficiently through seawater and match the sensitivity of many deep-sea eyes. For hunters, a small light can become bait. Certain deep-sea anglerfishes carry a modified spine ending in a luminous lure called an esca. In several groups, bacteria housed within this structure supply the glow, receiving a protected habitat while the fish gains a beacon capable of drawing curious animals toward its mouth. The surrounding water remains dark enough that a tiny point of radiance can become the most conspicuous object nearby. Other animals use light to become harder to see. Counter-illumination places luminous organs along the underside of the body. In depths where a trace of downwelling light remains, a fish or squid can adjust this glow until its belly resembles the faint brightness above. Viewed from beneath, the usual dark silhouette weakens. Camouflage here is achieved not by matching a surface, but by manufacturing the background. Defense can be brighter and more abrupt. Some crustaceans release luminous material into the water, leaving a glowing disturbance between themselves and an attacker. Certain squid produce radiant secretions. Other organisms flash when touched, potentially startling a predator or attracting a larger hunter that may attack the original threat. Light becomes a message whose recipient is not always the animal nearest to it. Communication demands greater precision. Patterns, positions, and timing of luminous organs can help animals distinguish members of their own species across darkness. In habitats where encounters may be infrequent, recognition matters. A signal that reaches the wrong animal wastes energy and may reveal the sender to something hungry. Evolution has even expanded the available palette. Most deep-sea creatures are relatively insensitive to red wavelengths, which seawater removes rapidly from sunlight near the surface. Yet, loose-jawed dragonfishes of the family Malacosteidae can produce red bioluminescence, and detect it. Specialized visual pigments and filtering structures allow these predators to illuminate nearby prey with light. Many neighboring animals perceive poorly, creating something resembling a private searchlight. The chemistry behind living light has appeared independently many times across the tree of life. Bacteria, dinoflagellates, jellyfish, comb jellies, crustaceans, squid, and fishes all contain luminous representatives, but they do not share a single ancestral lantern. Natural selection repeatedly arrived at light because darkness created opportunities for any organism able to generate, detect, or manipulate a photon. Within the deep Sargasso, these signals rarely form a continuous glow. They exist as scattered events across immense volumes of water, brief points awakened by movement, hunger, contact, or courtship. Human submersibles glimpse only fragments when their presence disturbs the surrounding animals. Beyond those observations, countless flashes occur without witnesses, exchanged between eyes and bodies shaped to interpret them. At these depths, darkness is therefore not an empty absence of vision. It is the background against which living signals acquire meaning. A blue pulse, a red beam, a luminous cloud, or the steady lure of bacteria can alter the outcome of an encounter, making chemistry itself part of the language of survival. Among the animals connected to the Sargasso Sea, the eel carries a mystery so old that its first written explanation belonged to a world without microscopes, oceanographic vessels, or any knowledge of migration across an ocean. For centuries, people could catch eels almost everywhere around the North Atlantic, yet the beginning of an eel's life remained absent from human sight. European eels inhabit waters from North Africa through much of Europe, reaching rivers and lakes as far north as Scandinavia. American eels occupy waters from Greenland and eastern Canada, south toward Florida and the Caribbean. Both can spend many years far from the open Atlantic, feeding and growing in estuaries, streams, lakes, and rivers. Their familiar presence made one absence especially puzzling. Eggs were not found in those freshwater habitats, and mature breeding adults seemed to disappear before reproduction could be observed. Around 350 before the Common Era, Aristotle confronted the same problem. Unable to identify reproductive organs or young emerging from eggs, he concluded that eels arose spontaneously from the earth itself. The idea sounds strange now, but it reflected the evidence available to an observer who could see adult eels appearing in waterways without ever witnessing their birth. More than 2,000 years later, the mystery still resisted examination. Before Sigmund Freud became associated with psychology, the young researcher worked at a marine zoological station in Trieste in 1876. He dissected hundreds of eels while searching for male reproductive anatomy. His specimens offered no satisfying answer. The reason lay within the animal's extraordinary life cycle. Sexual development becomes pronounced only as eels transform for their final ocean migration, long after many of the individuals encountered inland have been studied. The decisive clues came not from adult eels, but from their nearly transparent offspring. Danish biologist Johannes Schmidt began tracing peculiar, flattened larvae called leptocephali through the Atlantic. Rather than searching for a hidden nest, he treated larval size as a map. Larger larvae occurred nearer Europe, progressively smaller ones appeared farther west. If the youngest animals marked the region closest to spawning, their distribution could reveal the birthplace that adults had concealed. For years, Schmidt and his collaborators crossed enormous stretches of ocean, towing fine nets and measuring tiny specimens. By the early 20th century, the pattern had narrowed toward the western North Atlantic. the smallest European eel larvae repeatedly appeared within the Sargasso region. American eel larvae were also concentrated there, revealing that two species occupying opposite sides of an ocean returned to the same broad sea to reproduce. Their journeys are astonishingly different in length. An American eel may travel from continental waters into the western Atlantic over a comparatively shorter route. A European eel leaving a river in Norway, Ireland, France, or the Mediterranean faces a migration spanning thousands of miles. During this transformation, its body changes for ocean travel. The eyes enlarge, coloration shifts toward silver, digestive activity declines, and stored energy becomes the fuel for a journey from which the adult is not expected to return. Spawning itself remains remarkably difficult to witness. Scientists have collected ever younger larvae and used tracking technology to follow migrating adults toward the Sargasso, building powerful evidence for the destination. Yet direct observation of European eels mating in the open ocean has remained elusive. Their birthplace was inferred from traces scattered across water rather than from a witnessed spawning event. From those waters, transparent larvae begin drifting outward on Atlantic currents. Eventually, they transform into glass eels, approach continental coasts, and enter the waterways where generations of observers once assumed their lives began. The rivers were never their birthplace. They were only the long middle passage of a life whose beginning and ending belonged to the Sargasso Sea. By the time a European eel leaves fresh water, the animal that entered those rivers years earlier has begun to disappear. Its final transformation prepares it for a journey of roughly 3,700 miles across the Atlantic, toward a destination known from evidence accumulated around the travelers, rather than from anyone watching them complete it. For perhaps 5 to 20 years, an eel may have grown among European rivers, lakes, estuaries, and coastal waters. Then comes the silvering stage. Its skin develops a darker back and reflective flanks suited to the open ocean. The eyes enlarge dramatically, improving sensitivity under dim conditions. The pectoral fins change, energy reserves accumulate, and the digestive tract regresses. Feeding gives way to migration. Fat stored during years of growth must support both the crossing and the development of reproductive tissue. Autumn departures carry these silver eels downstream and through estuaries into the Atlantic. Once offshore, their movements become difficult to follow. Electronic tags have revealed that migrating eels do not simply travel along a flat surface route. They perform repeated vertical movements through the water column, often occupying deeper water during daylight and rising after dusk. Temperature, pressure, predators, navigation, and energy conservation may all influence this pattern, but the complete purpose remains unresolved. A study published in Scientific Reports in 2022 brought researchers closer than previous tracking efforts. Eels released and tagged in the Azores were recorded moving southwest toward the Sargasso Sea, providing direct evidence that adults were following the expected route into the presumed breeding region. Some reached the boundary of that broader spawning area, yet technology still did not accompany an eel through the reproductive event itself. That missing observation leaves an unusual gap between what biology strongly supports and what human eyes have actually witnessed. Larval distributions indicate that spawning occurs across a broad region of the Sargasso. By examining the ages, sizes, and locations of the youngest larvae, researchers can work backward through their drift. Seasonal patterns point towards spawning during winter and spring, with activity thought to peak around late February. Navigation remains another unresolved part of the crossing. An eel leaving a European watershed has no previous adult migration to remember. Its route must therefore emerge from inherited sensory abilities. Experiments suggest that eels can respond to Earth's magnetic field, raising the possibility of an internal magnetic compass. Ocean currents, temperature boundaries, pressure changes, odors, and other environmental cues may contribute as well. None has yet supplied a complete navigational explanation for a voyage spanning an ocean. Even the final hours are reconstructed indirectly. European eels are believed to mature fully near the spawning grounds, release eggs and sperm into open water, reproduce once, and die. This pattern fits their physiology and the life history of related species. Yet wild European eel eggs have never been conclusively collected from the spawning grounds, and the bodies of adults completing reproduction have not provided the direct scene researchers have sought. The scale of that absence is remarkable. Countless eels have inhabited waterways beside cities, farms, forests, ports, and villages for centuries. Humans have trapped them, eaten them, studied their tissues, tagged their bodies, and followed portions of their migration across the Atlantic. Still, the act around which their entire life cycle is organized remains beyond direct observation. Each generation, therefore, ends where the next becomes detectable only later, as transparent larvae moving away through ocean currents. Between the arriving adults and those first drifting young lies a brief biological event concealed within an immense sea, supported by layers of evidence, yet never directly witnessed. An eel beginning life in the Sargasso Sea possesses no map in any familiar sense. It has no parent beside it, no learned route, and no memory of a river waiting beyond the Atlantic. Yet, its life will eventually connect two places separated by thousands of miles, with the return journey beginning years after the outward passage that carried it toward Europe. As a transparent leptocephalous larva, the young European eel is transported eastward within the North Atlantic circulation. The crossing can occupy years of early development. By the time it approaches Europe and North Africa, its body has transformed into the nearly transparent glass eel stage. Coastal water introduces an entirely new chemical landscape. Many individuals enter estuaries and rivers, gradually becoming pigmented young eels and dispersing inland into habitats their ancestors may have used generations before. There they can remain for years, sometimes approaching 2 decades or longer, without making the oceanic migration themselves. Eventually, silvering changes the body for departure. What follows presents a navigational problem of unusual scale. An eel leaving the Rhine, the Thames, or another European watershed must first reach the sea, then move generally southwestward through an Atlantic filled with shifting currents, storms, temperature boundaries, and few fixed landmarks. One possibility lies in a force passing through the eel and the planet alike. Earth's magnetic field varies geographically in strength and orientation, producing patterns that some migratory animals can detect. Laboratory experiments indicate that European eels respond to manipulated magnetic conditions. Juveniles exposed to magnetic fields, representing different geographic locations, have altered their swimming direction, evidence that magnetic information can influence orientation. From this emerged the idea of a magnetic map. A larva traveling away from its birthplace might acquire information associated with the magnetic character of the region, later using related cues during its adult return. Such imprinting would offer an elegant explanation for navigation without teaching or previous adult experience. Yet, detecting a magnetic field is not the same as demonstrating a complete map across roughly 3,700 miles of ocean. The sensory structures involved remain uncertain, and experiments cannot yet reconstruct the entire migration. Currents complicate the picture further. Eels do not travel through motionless water. The Atlantic itself transports heat and water in broad moving systems, potentially assisting, redirecting, or resisting a swimmer. Some researchers have therefore considered whether successful migration depends partly on entering favorable currents rather than following a precise internal route from beginning to end. Navigation and physical transport need not be opposites. An animal may orient while allowing moving water to perform part of the journey. Even the destination has attracted renewed scrutiny. In 2020, researchers proposed that spawning might occur nearer the mid-Atlantic ridge, challenging the traditional interpretation built from Johannes Schmidt's larval surveys. The proposal stimulated debate, but subsequent examination found no corresponding concentration of the youngest larvae there. Evidence continues to support the Sargasso region as the reproductive center, while the exact movements of arriving adults remain difficult to document. Technology has narrowed the unknown without removing it. Electronic tags can reveal direction, depth, temperature, and portions of individual routes. Genetic analysis can connect larvae with populations spread across Europe. Controlled magnetic experiments can isolate one sensory cue at a time. Oceanographic models can estimate how currents influence movement. None has yet produced a complete chain from an inland adult to a witnessed arrival at its breeding ground. The puzzle therefore survives in a more precise form than Aristotle could have imagined. Science knows the broad destination, measures pieces of the route, and identifies senses that may contribute. What remains hidden is how those pieces become a journey inside the animal itself, allowing an eel raised far from its birthplace to orient across an ocean toward waters it encountered only at the beginning of life. The first body of an eel is built for drifting rather than swimming against an ocean. After hatching in the Sargasso Sea, the larva enters the water as something so unlike the familiar adult that generations of naturalists placed the two forms in separate branches of their catalogs. Its body is flattened from side to side, almost leaf-like, with tissues so transparent that the surrounding water seems to pass through it. This larval form is called a leptocephalus, a name derived from Greek words referring to a thin head. Early specimens offered few clues about their future. They possessed delicate bodies, elongated teeth, and little of the muscular cylindrical shape associated with eels. European zoologists eventually described some of them as a separate species called Leptocephalus brevirostris. The mistake endured because no observer had watched the apparent species become anything else. That changed through the work of Italian zoologist Giovanni Battista Grassi and his colleagues during the late 19th century. In 1896, Grassi reported the developmental connection between leptocephaly and young eels, helping resolve a biological confusion that had survived for generations. The strange, transparent fish was not an obscure adult species. It was an eel before an eel looked like one. Its unusual anatomy reflects an equally unusual period of life. A leptocephalus contains a gelatinous interior, rich in compounds that help support its thin body with relatively little dense tissue. This construction reduces the energy needed to remain suspended in open water. Instead of behaving like a miniature version of the adult, the larva is adapted to a prolonged pelagic existence, growing while currents transport it across the Atlantic. Feeding during this stage remains less straightforward than the feeding of most familiar fish larvae. Researchers have found evidence that leptocephaly consume tiny particles of organic material suspended in seawater, including marine snow, mucus structures, discarded by planktonic animals and other soft detrital matter. Their long larval teeth disappear during metamorphosis, emphasizing how completely the feeding apparatus is rebuilt as the animal approaches its next life stage. European eel larvae begin only a fraction of an inch long. During their oceanic development, they lengthen gradually until larger leptocephaly may approach 3 inches. Growth does not simply continue into adulthood. Near the continental margin, the leaf-like body begins a remarkable reconstruction. The larva becomes shorter as its proportions change, the head and jaws reorganize, and the broad, transparent profile contracts into the narrow, cylindrical form of a glass eel. Transparency remains for a time. A newly transformed glass eel can be almost colorless, its internal structures visible through the body. Pigment develops later as it approaches estuaries and coastal habitats. What began as a drifting leaf has become a swimmer prepared for an entirely different environment. This transformation is repeated across immense numbers of larvae released into the North Atlantic system. European eel leptocephaly are carried generally eastward and northeastward toward Europe and North Africa, while American eel larvae are transported toward western Atlantic coasts. Currents provide much of the motion, sorting developing animals across an ocean basin while the larvae themselves grow and change within the moving water. The connection reaches far beyond the eel. Atlantic circulation transports heat toward northwestern Europe, while also carrying living passengers whose entire continental population depends upon that journey. Each eel entering a distant estuary arrives only because an almost invisible larval body survived months of feeding, drifting, predation, and transformation in open water. For centuries, that transparent beginning concealed the animal's identity from science. Its form was not merely unfamiliar. It belonged to a life stage so different from adulthood that recognizing the same creature required watching one body become another. At the beginning of its Atlantic crossing, the European eel possesses almost no authority over direction. Barely 1/8 of an inch long after hatching, it enters a moving ocean whose currents operate on scales far beyond its own strength. Its first great journey is therefore written less by muscle than by water, through a circulation system linking the western subtropics with distant European shores. Along the western boundary of the North Atlantic, the Gulf Stream gathers warm water into a swift northeastward flow. Near the Florida Straits, this current transports roughly 8 billion gallons every second, although its volume grows farther downstream as surrounding waters join it. The current follows the eastern margin of North America before bending away from the continent near Cape Hatteras. Beyond that turn, its influence continues into the broader North Atlantic circulation, creating pathways through which drifting organisms can travel enormous distances. Eel larvae enter this moving network at an exceptionally vulnerable stage. They are capable of swimming, and evidence suggests their behavior can influence depth and orientation, but they cannot simply overpower the basin-scale circulation carrying them. Their vertical position matters because currents change with depth. By occupying different layers, even a small larva may encounter water moving at another speed or angle, allowing behavior and ocean physics to work together rather than reducing the journey to passive drift. The two Atlantic freshwater eel species eventually separate across this circulating landscape. American eel larvae are recruited toward the western Atlantic, reaching the Caribbean, Gulf of Mexico, eastern North America, and other connected coastal regions. European eel larvae remain within eastward transport pathways much longer. Their route can extend across roughly 3,700 miles of ocean, and estimates of larval duration vary, with the crossing commonly understood to require many months and potentially several years. By the time European juveniles approach the continental shelf, the drifting phase has prepared them for a different kind of movement. Glass eels arriving near estuaries can swim actively, respond to tides, and use incoming water to advance toward coastal and freshwater habitats. Some continue far upriver. Others remain in estuaries or brackish lagoons. The species is remarkably flexible in where it grows, even though every individual ultimately traces its origin back across the Atlantic. There, during the yellow eel stage, growth may continue for years or decades. Feeding replaces drifting as the dominant task. Body mass increases, energy is stored, and the animal becomes part of local food webs thousands of miles from its birthplace. Yet, the oceanic connection remains embedded in the population. No European river produces its own independent lineage. Recruitment depends upon larvae successfully crossing the Atlantic and reaching continental waters from a shared spawning region. That dependence has become increasingly important as European eel numbers have fallen. Recruitment of young eels to Europe declined dramatically from levels recorded during the late 20th century, with some long-term monitoring series showing losses exceeding 90%. Fishing pressure, barriers such as dams, turbine mortality, habitat alteration, pollution, parasites, and changes in ocean conditions have all been investigated as contributors. No single explanation accounts comfortably for the entire decline. The Gulf Stream connection, therefore, carries more than larvae. It links distant habitats into one biological system, joining subtropical spawning waters with estuaries, wetlands, lakes, and rivers scattered across Europe and North Africa. An eel may spend most of its visible life inland, beneath reeds or riverbanks, yet its arrival depended on currents crossing an ocean before it had ever touched fresh water. For every generation, that immense ribbon of moving Atlantic water remains part of the animal's inheritance, carrying new life between worlds, separated by thousands of miles. A bottle cap can cross an ocean without ever choosing a direction. Once it reaches the moving waters of the North Atlantic, wind and current begin deciding its path, carrying it away from shore and into the broad circulation surrounding the Sargasso Sea. The same physics that gathers drifting sargassum can gather human debris, concentrating objects released thousands of miles apart into overlapping routes across the open water. Much of that material begins far inland. Rain washes discarded packaging into drains and streams. Rivers deliver fragments to estuaries, while coastal cities, shipping, fishing, and offshore activity add other sources directly to the sea. From eastern North America, Western Europe, the Caribbean, and Western Africa, floating debris can enter current systems connected to the North Atlantic subtropical gyre. Wind pushes especially buoyant objects across the surface, while waves repeatedly immerse and release them, slowly altering the course of each piece. Once inside the rotating circulation, escape may take years. The gyre is not a solid wall, and its water is constantly moving, but its converging surface currents favor accumulation within the subtropical interior. Oceanographers describe these regions as convergent zones, places where floating material tends to become concentrated because large-scale circulation carries surface water inward. Sargassum responds to the same forces which brings natural habitat and synthetic debris together. That overlap changes the meaning of a floating mat. Among the branching weed can be bottle caps, rope fibers, fragments of containers, pieces of fishing equipment, disposable lighters, footwear, toys, and objects whose original purpose has become difficult to recognize. During one Greenpeace expedition, researchers examining a comparatively small sargassum mat collected more than 300 pieces of plastic in roughly half an hour. The debris had become physically woven into habitat used by animals for shelter, feeding, and development. Large objects, however, represent only the most visible stage of the process. Sunlight weakens exposed plastic through ultraviolet radiation. Waves bend and abrade it. Repeated collisions fracture brittle surfaces into progressively smaller pieces. Plastic does not need to dissolve to disappear from ordinary sight. It can simply become fragmented until a recognizable bottle, crate, or line has been reduced to particles scattered through the water. Some fragments remain near the surface because their polymers are less dense than seawater. Others acquire films of bacteria, algae, and small organisms, a process known as biofouling. Added biological material can increase their density enough to make them sink. If that coating is later consumed or decomposes, buoyancy may change again. Individual particles can therefore move vertically through the water column, linking the floating accumulation above with deeper ecosystems beneath it. This makes the Sargasso's plastic problem larger than the debris visible between strands of weed. Microplastics can occur throughout seawater and can be encountered by organisms small enough to mistake particles for food or ingest them alongside natural material. Fibers and fragments also provide hard surfaces where microbes and tiny animals can settle, creating artificial substrates in an environment where solid surfaces are otherwise scarce. The gyre has therefore become an unintended archive of Atlantic consumption. Objects manufactured for minutes, days, or years can remain within marine circulation far longer than their useful lives on land. Their labels fade, their colors bleach, and their edges become rounded, yet the material persists as it is divided into smaller forms. At the surface, accumulation can still be counted piece by piece. Beneath it, that accounting becomes harder. The plastic is no longer merely floating beside the ecosystem. Through fragmentation, fouling, sinking, ingestion, and transport, it has entered the water column itself, dispersed among the living particles of the Sargasso Sea. Seen from beneath the surface, plastic loses the familiar shapes that once revealed what it was. A bottle no longer needs to remain a bottle, and a bag no longer needs to resemble a bag. Given enough sunlight, motion, and time, each can become a cloud of fragments, small enough to drift among plankton. In the Sargasso Sea, researchers found those fragments suspended through water that appeared almost impossibly clear. Microplastics are generally defined as plastic particles smaller than roughly 1/5 of an inch. Some enter the ocean already microscopic, while many begin as larger objects that gradually fracture. Ultraviolet radiation from sunlight weakens polymer bonds near exposed surfaces. Waves flex brittle material, collisions scrape away edges, and years of weathering divide one discarded object into hundreds or thousands of pieces. Fragmentation changes the size of the pollution without removing the material itself. During Greenpeace investigations in the Sargasso, scientists collected water samples containing extraordinary numbers of these particles. One sample held nearly 1,300 fragments. Analysis conducted with researchers associated with the Five Gyres Institute and the Scripps Institution of Oceanography placed concentrations in parts of the North Atlantic accumulation zone within the same broad scale observed in heavily polluted regions of the North Pacific. The phrase garbage patch can create a misleading picture. There is no continuous island of bottles and bags stretching across the sea. Much of the accumulation is dispersed, sometimes nearly invisible from a vessel. A container lowered into apparently clean water can reveal what the eye misses: tiny fibers, chips, films, and irregular grains mixed into the same moving layer occupied by drifting organisms. Their small size gives these particles access to biological pathways unavailable to larger debris. Copepods and other planktonic animals can encounter particles within the size range of natural food. Small fish may consume contaminated prey or ingest fragments directly. Predators then feed upon those animals. The result is not a simple ladder in which every particle inevitably climbs upward, but a network of possible transfers through digestive systems, waste, carcasses, and sinking organic matter. Plastic surfaces also change after entering seawater. Microorganisms colonize them, forming thin biological coatings that can alter odor, density, and chemical behavior. To an animal searching through suspended particles, a weathered fragment covered with marine life may no longer behave like an obviously foreign object. The distinction between manufactured debris and edible material becomes difficult at the scale where feeding decisions occur in fractions of a second. Much of the material reaching the ocean comes from products designed for short periods of use: packaging, containers, synthetic fibers, and fishing materials can persist long after their original purpose has ended. Globally, only a small fraction of plastic waste has historically been recycled. Large quantities have instead been buried, burned, discarded, or lost into environments where rivers and coastal waters provide routes toward the sea. Within the North Atlantic circulation, those scattered losses can converge. The Sargasso does not need a shoreline to accumulate the residue of distant cities. Its geography is written in moving water, and that movement can gather fragments from widely separated sources into the same broad region. This makes microplastic pollution especially difficult to perceive. The largest debris can be photographed, counted, and removed piece by piece. The smallest material has already crossed another boundary. It has become part of the suspended texture of the ocean, occupying the spaces between living cells, drifting larvae, organic particles, and grains of marine snow. In water renowned for clarity, human material can remain almost unseen precisely because it has been broken into pieces small enough to disappear within the sea itself. A drifting loggerhead hatchling may weigh only a few ounces, yet the floating world around it already contains choices its ancestors never faced. Among the branching sargassum are crustaceans, fish eggs, bits of organic matter, and weathered plastic fragments similar in size to natural food. The pollution is no longer merely passing beside the habitat. It has entered the physical structure of the nursery itself. Surveys of sargassum accumulation zones have repeatedly recovered plastic entangled among the weed. Fibers catch around narrow branches. Small chips settle into dense mats. Larger pieces become surfaces where algae, bacteria, and tiny invertebrates grow. Once coated with marine life, a fragment can acquire odors and textures associated with food, making the boundary between natural material and manufactured debris less obvious to an inexperienced feeder. Young sea turtles are especially exposed because floating sargassum provides shelter and feeding opportunities during vulnerable early life. Studies of juvenile turtles from the North Atlantic have documented frequent ingestion of plastic, including fragments small enough to pass into the digestive tract. Plastic can occupy space without supplying nutrition, irritate tissues, or contribute to blockage when larger pieces accumulate. The outcome depends on particle size, quantity, polymer type, and the condition of the animal. Small fishes, living around Sargassum encounter the same altered menu. Juveniles of mahi-mahi, jacks, triggerfish, and other open ocean species hunt among the weed, taking crustaceans and smaller animals that may themselves have encountered microplastics. Exposure can therefore occur directly through mistaken ingestion or indirectly through prey. Laboratory research across marine species has linked high microplastic exposure to effects on feeding, growth, inflammation, and energy use, although translating those experiments into exact consequences for wild Sargasso populations remains difficult. For eel larvae, the concern carries another dimension. Their early development unfolds within Atlantic waters, now containing synthetic particles that were absent through most of their evolutionary history. Leptocephali feed differently from many familiar fish larvae, taking in suspended and gelatinous organic material rather than simply hunting visible prey. That places them within the same drifting particle field where microscopic plastic can occur, even though the precise biological effects on wild European and American eel larvae remain incompletely understood. Plastic can also interact with chemicals. Weathered surfaces may attract persistent organic pollutants from surrounding seawater, including certain pesticides and polychlorinated biphenyl compounds. Additives incorporated during manufacturing can also be present within the plastic itself. After ingestion, some chemicals may transfer between the particle and an animal's tissues. But the importance of plastic as a chemical carrier varies greatly and can be smaller than exposure from contaminated food or water. The chemistry is real, while its ecological contribution depends on circumstance. What begins in a floating mat can therefore intersect with many feeding relationships. A small crustacean may encounter a fiber. A fish may eat that crustacean. Larger predators may consume the fish, while whales, seabirds, turtles, and people occupy different branches of the wider Atlantic food web. Microplastics do not necessarily move upward in a simple, ever-increasing sequence, because animals can excrete particles, and different species retain them differently. Their presence nevertheless creates repeated opportunities for contact across many levels of life. The deeper change is ecological rather than visual. Sargassum still gathers into golden rafts, sheltering young animals as it has for generations. Yet woven through that living architecture are materials produced far beyond the horizon, carried by rivers, coastlines, ships, and currents into a habitat with no boundary separating pollution from biology. For the animals growing there, plastic has become another particle in the water, another surface in the weed, and another substance their bodies must somehow negotiate. Plastic does not need to remain in one place to be trapped. Inside the North Atlantic Gyre, confinement is a matter of circulation. Water moves continuously, yet much of that movement bends back into the same immense rotating system, carrying suspended debris through paths that can keep it within the region for decades. Oceanographic models have estimated residence times reaching roughly 50 years, turning the Sargasso into a long-lived reservoir for material that arrives from far beyond its boundaries. That timescale changes the meaning of every discarded fragment. Plastic entering the circulation today may remain within the gyre into the 2070s, while material released during the 1970s can still persist as altered remnants. Bottles, packaging, rope, fishing equipment, and countless smaller objects rarely preserve their original shapes for that entire journey. Sunlight weakens exposed surfaces, waves flex brittle polymers. Salt, heat, and mechanical abrasion continue the process until recognizable objects become flakes, fibers, and particles. This breakdown is not the same as biological decay. Wood, leaves, and animal remains can be dismantled chemically and recycled through living systems. Conventional plastics behave differently. Many resist the natural processes that would return organic matter to simpler biological ingredients. Instead of vanishing, they fragment. A piece visible from a boat can eventually become grains, difficult to distinguish from suspended matter, then particles smaller still, extending contamination into dimensions where collection becomes increasingly difficult. Some of those particles remain near the surface, but others do not. Their movement depends on density, shape, biological growth, turbulence, and interactions with organic material. Microorganisms can colonize plastic and change its buoyancy. Particles may sink, become incorporated into marine snow, descend through the water, or later move again as conditions change. The pollution, therefore, occupies more than the bright upper skin of the sea. Its persistence is measured not only across distance, but through depth and generations. Scale makes removal especially difficult. The Sargasso Sea spans roughly 1,200,000 square miles, without a coastline where debris can simply be intercepted and gathered. Even where floating plastic becomes concentrated, nets capable of catching the smallest fragments would also capture plankton and other organisms. Below the surface, the challenge expands through thousands of feet of water. Recovering dispersed microscopic particles without disturbing the life surrounding them remains beyond any practical large-scale technology now available. At sufficiently small sizes, plastic can interact with organisms in ways larger debris cannot. Laboratory studies have shown that microscopic and nanoscopic particles can cross biological barriers in some species, reaching tissues beyond the digestive tract under particular exposure conditions. The extent of this process in wild Sargasso organisms remains an active field of research, but fragmentation steadily increases the number of particles available for contact. One object can become thousands of smaller pieces, multiplying surfaces and pathways through the ecosystem. Time deepens that transformation. Industrial plastic production expanded rapidly after the middle of the 20th century, meaning the ocean has received most of this synthetic material within a fraction of an ecological lifetime. The Sargasso system itself was shaped across geological ages by currents, climate, evolution, migration, and the slow accumulation of biological relationships. Against that history, widespread plastic contamination arrived almost instantaneously. The gyre now performs the same physical work it always has. Currents circle. Floating material converges. Water remains enclosed for long intervals. Nothing in that ancient circulation distinguishes a seed, a drifting organism, or a weathered polymer fragment. The mechanism that gathers life also retains what human activity has added, and each year of continued input joins material already circulating from decades before. Isolation does not make the Sargasso Sea empty. It makes every exchange unusually revealing. Far from continental shelves and major river inputs, its central waters receive few of the nutrients that fertilize coastal ecosystems. Nitrogen and phosphorus can become scarce, yet microscopic life persists in immense numbers. For oceanographers, this apparent contradiction has made the region a natural laboratory for discovering how marine ecosystems function when almost everything useful must be captured, recycled, and used with extraordinary efficiency. The surrounding currents do not seal the Sargasso completely, but they restrict and organize exchange enough to create distinctive conditions over broad areas. Water masses can remain within the subtropical circulation for long periods, allowing researchers to follow changes in chemistry, plankton, carbon, and microbial communities without the constant influence of nearby land. Long-running observations near Bermuda have therefore become especially valuable, revealing seasonal and multi-year changes in an ocean region once assumed to be comparatively uniform. Among the most important inhabitants are organisms too small to see individually. Bacteria belonging to the group known as Pelagibacterales, historically called the SAR-11 clade, rank among the most abundant cellular organisms in the ocean. Their cells are remarkably small and streamlined, carrying compact genomes suited to waters where nutrients occur in minute concentrations. Rather than wasting energy maintaining unnecessary biological machinery, they specialize in extracting dissolved organic compounds that other organisms release into the surrounding sea. Another microscopic resident transformed scientists' understanding of photosynthesis. Prochlorococcus, discovered during research involving samples from the Sargasso region in the 1980s, is among the smallest known photosynthetic organisms. Individual cells are almost unimaginably tiny, but collectively their relatives contribute substantially to marine primary production. Their success demonstrated that nutrient-poor open water could support enormous photosynthetic activity through organisms operating at scales traditional sampling methods had often overlooked. The hidden diversity became clearer in 2004 when researchers led by geneticist Craig Venter published a large environmental genome survey based on Sargasso Sea samples. Instead of attempting to grow each microorganism in a laboratory, the team extracted genetic material directly from seawater and analyzed the mixture. The study uncovered more than 1 million previously unknown genes and evidence for at least 1,800 genomic species, revealing biological complexity that conventional cultivation had failed to capture. That approach, known as metagenomics, changed marine microbiology. A bottle of seawater could contain a genetic archive representing thousands of organisms, many belonging to lineages scientists had never cultured or named. Their genes revealed biochemical strategies for gathering nutrients, responding to sunlight, processing carbon, and surviving periods of scarcity. The apparently clear water of the Sargasso was not biologically simple. Much of its complexity merely existed below the resolution of ordinary vision. Its scarcity of nutrients makes those discoveries globally relevant. Microbes repeatedly recycle nitrogen, phosphorus, and organic carbon before those materials can sink away, allowing productivity to continue in water that offers little fresh nourishment. Tiny photosynthetic cells capture solar energy, grazers consume them, bacteria process released compounds, and nutrients return to circulation through an intricate microbial loop. Efficiency, rather than abundance of raw resources, sustains much of this ecosystem. As ocean temperatures rise and seawater chemistry changes, subtropical nutrient-poor regions are expected to remain important indicators of marine change. Decades of measurements from the Sargasso provide researchers with something increasingly precious, a long record against which new conditions can be compared. Within its slowly circulating waters, microscopic organisms preserve evidence of how life responds when nutrients are scarce, temperatures shift, oxygen changes, and carbon chemistry moves away from patterns that shaped earlier generations. A single bottle of seawater can describe a moment. A sequence of bottles collected from the same ocean for decades can reveal something larger, the gradual movement of the ocean itself through time. In October of 1988, Researchers traveled roughly 50 miles southeast of Bermuda and began returning to a fixed region of the Sargasso Sea near 31 degrees, 40 minutes north, and 64 degrees, 10 minutes west. Their purpose was simple in design and immense in duration: measure the same water repeatedly with methods precise enough to make distant years comparable. The project became the Bermuda Atlantic Time Series study, operated through the Bermuda Institute of Ocean Sciences. Research cruises returned approximately every month, lowering instruments through the water column and collecting samples at different depths. Temperature, salinity, dissolved oxygen, nutrients, carbon chemistry, plankton, and biological productivity were recorded again and again. Each visit produced another narrow slice of the Sargasso, and those slices gradually assembled into one of the longest sustained records of open ocean change on Earth. Its power comes from repetition. A warm month can be weather, an unusual plankton bloom can be seasonal variation. A temporary change in oxygen can disappear before the next cruise. But when observations continue across more than 35 years, patterns begin separating from temporary disturbances. Seasonal cycles become recognizable against their own history, and slow changes that would remain invisible during a short expedition emerge from thousands of measurements. The record has shown that the upper ocean near Bermuda is not fixed. Temperatures vary across seasons and years, while longer trends accumulate beneath those fluctuations. Biological communities respond to changes in mixing, nutrient availability, and water structure. Winter conditions can stir nutrients upward from deeper layers, influencing productivity during subsequent months. Differences in the strength and depth of that mixing can ripple through microscopic communities, leaving signatures that become meaningful only when compared with decades of earlier observations. Carbon chemistry has provided one of the clearest long-term signals. Human activity releases carbon dioxide into the atmosphere, and the ocean absorbs a substantial portion of those emissions. Once dissolved in seawater, carbon dioxide participates in chemical reactions that increase acidity and reduce the availability of carbonate ions used by many organisms to construct shells and skeletons. Measurements near Bermuda have documented this progressive alteration of seawater chemistry across decades, helping establish open ocean acidification as a measurable process rather than a distant theoretical expectation. Such detection requires consistency. Instruments improve, laboratory techniques evolve, and research teams change. Yet long records depend on careful calibration, so measurements collected decades apart remain scientifically comparable. The value lies not in any individual cruise, but in preserving continuity through generations of researchers. A sample gathered in 1989 becomes more informative when placed beside another collected in 2010, then another from 2025. Location matters as much as duration. Far from major rivers and densely populated coastlines, the central Sargasso offers researchers a clearer view of broad ocean processes without many immediate terrestrial influences. Changes arriving through the atmosphere, circulation, deep mixing, and global carbon chemistry can be followed against a comparatively stable geographic background. After more than 3 decades, the study has become something its founders could only create by waiting. Every month added another point to a record whose scientific value increased with age. The water passing beneath the research vessel never returned in exactly the same state, yet the measurements returned to the same place. Across those repeated visits, gradual planetary change became visible not as a single dramatic event, but as a persistent alteration written into temperature, chemistry, oxygen, nutrients, and living cells. The change does not arrive as a single warm season. It appears as a slope drawn through decades of measurements, small differences accumulating until the direction becomes unmistakable. In the Sargasso Sea, the long record from the Bermuda Atlantic Time Series study, supported by satellite observations across the wider basin, shows an ocean whose upper waters have been warming. The measurements belong to different years, different cruises, and different instruments, yet together they reveal a physical environment gradually departing from the conditions recorded when systematic monitoring began. Heat changes more than temperature. As surface water warms, it becomes more buoyant relative to the colder water below, strengthening the density boundary between layers. This increased stratification makes vertical mixing more difficult. Nutrients stored at depth are then less readily returned to sunlit surface waters, where microscopic photosynthetic organisms depend upon them. In a sea already known for low nutrient concentrations, a stronger barrier between the illuminated surface and the reservoir below can further limit biological production. The effect travels downward through the ecosystem. Phytoplankton form the foundation of open ocean food webs, transforming sunlight and dissolved carbon dioxide into organic matter. When nutrient supply changes, the consequences can pass through grazers, fish, drifting larvae, and the organisms that depend upon sinking material from above. Long records are especially valuable here because natural cycles can obscure gradual trends. A productive year does not erase warming, just as an unusually cool season does not reverse a change unfolding across decades. Oxygen carries another temperature signal. Warmer seawater can hold less dissolved oxygen, while stronger stratification can reduce the ventilation that replenishes deeper layers. Observations across the world's oceans have documented declining oxygen in many regions, and changes in the North Atlantic are part of that larger concern. For animals occupying deeper water, oxygen availability helps define where metabolism can be sustained. Altering that distribution can reshape usable habitat even when the ocean above appears unchanged. At the same time, the chemistry is shifting. The Sargasso has absorbed carbon dioxide added to the atmosphere by human activity. Dissolved carbon dioxide changes the balance of seawater chemistry, lowering its pH and reducing carbonate ion availability. Calcifying organisms must build within those altered conditions. Pteropods form delicate shells of calcium carbonate, while coccolithophores surround themselves with microscopic calcium carbonate plates. Their responses vary among species and circumstances, but the chemical environment in which those structures are produced is measurably changing. For the European eel, the implications are harder to isolate. Its reproductive biology in the Sargasso remains largely beyond direct observation, so scientists cannot simply compare modern spawning behavior with a detailed historical baseline. Temperature can influence development, metabolism, currents, and the timing of biological processes, while changes in plankton communities may alter conditions encountered by larvae. These pressures arrive alongside fishing, barriers in rivers, parasites, pollution, habitat alteration, and other forces already associated with the species' decline. Assigning a precise share of that decline to Sargasso warming remains beyond the evidence. That uncertainty does not make the physical changes uncertain. More than 35 years of repeated measurements near Bermuda have recorded changes in temperature and carbon chemistry directly in seawater. Satellites extend the view across the surrounding Atlantic, while laboratory analyses reveal shifts invisible from the surface. The significance lies in their convergence. Heat, stratification, oxygen, acidity, and biology are connected parts of the same moving system. The Sargasso has always varied from season to season and year to year. Now its natural rhythms unfold upon a changing baseline, one measured patiently enough for the difference to become legible across the passing decades themselves. Beginning in the tropical Atlantic, the change first became visible not as a disappearance of sargassum, but as an extraordinary excess of it. Around 2011, satellite observations began revealing immense accumulations of floating brown algae outside the familiar Sargasso Sea. They gathered across waters between West Africa, the Caribbean, and the Gulf of Mexico, forming a recurring transatlantic feature that scientists came to call the Great Atlantic Sargassum Belt. At its largest, this belt has extended for more than 5,000 miles across the Atlantic. Individual patches merge into sprawling surface aggregations that can be detected from orbit, their abundance changing with seasons, currents, winds, and nutrient availability. The principal floating forms are Sargassum natans and Sargassum fluitans, relatives of the algae long associated with the Sargasso ecosystem. Here, however, their growth occurs on a scale capable of transforming entire coastlines. The causes are complex. Researchers have linked the expansion to a combination of ocean circulation, favorable temperatures, and increased nutrient availability. Rivers can deliver nitrogen and phosphorus from soils, fertilizers, sewage, and disturbed landscapes into coastal waters, while atmospheric deposition and ocean mixing can supply nutrients from other sources. The Amazon basin has received particular scientific attention, although researchers continue examining how river discharge, upwelling near West Africa, changing currents, and climate variability interact. No single source explains every year of exceptional growth. While floating offshore, sargassum can still provide habitat. Trouble develops when enormous quantities are driven toward land. Across parts of the Caribbean, Mexico, Florida, and neighboring coasts, windrows of algae have accumulated along beaches in volumes far beyond ordinary seasonal strandings. Fresh arrivals pile over older layers, creating dense banks that can reach several feet deep. Turtle nesting areas may become obstructed, and hatchlings can face an altered route between sand and open water. Nearshore ecosystems experience another set of pressures. Thick accumulations reduce the sunlight, reaching seagrasses and shallow benthic communities. As submerged sargassum decays, microorganisms consume dissolved oxygen, sometimes creating oxygen-depleted water around reefs, lagoons, and sheltered bays. Organic matter can alter local chemistry as decomposition proceeds. A plant that supports life while drifting offshore can therefore become an ecological burden when concentrated beyond the capacity of a coastal system to process it. On land, decomposition produces hydrogen sulfide, the gas responsible for the characteristic odor of rotten eggs. At elevated concentrations, exposure can irritate the eyes and respiratory system, while people with certain respiratory conditions may be especially vulnerable. Coastal communities must remove enormous masses before decay advances, yet collection itself is difficult. Heavy machinery can damage beaches, disturb nests, and remove sand along with the algae. Material collected after landfall may also contain salt, plastics, or other contaminants that complicate disposal and reuse. The economic effects follow the shoreline. Fishing boats can struggle through clogged shallows, waterfront businesses face repeated cleanup costs, and tourism destinations can lose usable beach space during severe arrivals. Communities that once treated stranded sargassum as an occasional natural event now plan for recurring seasons of collection, transport, monitoring, and disposal. Satellite records have transformed those local scenes into a basin-wide picture. What appears on one beach is part of a biological event spanning thousands of miles, shaped by currents that continually rearrange its boundaries. Its modern scale has no clear counterpart in the satellite era, making each season part of an unfolding record rather than a return to a well-documented historical norm. The Great Atlantic Sargassum Belt shows how the same floating algae can occupy two very different ecological roles. Dispersed at sea, it creates structure and shelter. Concentrated along developed and sensitive shores, its abundance changes water, air, habitat, and human activity together. A strand of sargassum has no fixed ecological meaning. Its effect depends on where currents carry it, how densely it gathers, and what surrounds it when it arrives. Suspended across the open Atlantic in scattered rafts, its branching structure creates habitat in water otherwise lacking solid shelter. Packed against a tropical shoreline in enormous masses, that same architecture can become a barrier between coastal life and the conditions it requires. Within offshore mats, the spaces among gas-filled floats and narrow fronds provide refuge for small animals. Juvenile fish weave through them, young sea turtles rest among them, and specialized creatures spend much of their lives within this drifting habitat. More than 120 species of fish have been associated with sargassum communities, alongside crabs, shrimp, snails, worms, and other invertebrates. Food, shade, attachment surfaces, and protection can all exist within a relatively small patch. Abundance changes those relationships. When currents and winds drive immense tropical blooms toward shore, separate patches compress into thick accumulations. Sunlight that once penetrated shallow water is intercepted near the surface. Beneath the floating layer, seagrasses receive less energy for photosynthesis, while corals and algae living on the seafloor experience altered light conditions. The physical weight of stranded material can cover intertidal habitat and reshape the narrow boundary where land and sea meet. Decomposition adds another pressure. Once large quantities begin dying, bacteria consume the organic matter and use dissolved oxygen in the process. In poorly flushed bays and lagoons, oxygen concentrations can fall enough to stress or kill fish and invertebrates. Decay also changes local water chemistry and can release hydrogen sulfide and ammonium. What had been living habitat offshore becomes a concentrated source of biological demand when enormous volumes break down together. Sea turtles encounter both sides of this paradox during a single lifetime. Young turtles may use floating sargassum as shelter during their early oceanic years, finding food and structure far from land. Females later return to sandy coasts to nest, where deep deposits of stranded weed can obstruct access to suitable sand. Newly emerged hatchlings face the reverse route. Dense banks between nests and water can slow them, trap them, or force them across longer stretches of exposed beach. The distinction is therefore not between useful sargassum and harmful sargassum. It lies in concentration, location, timing, and ecological capacity. Coastal systems can process ordinary amounts of naturally stranded vegetation. Extraordinary arrivals can exceed that capacity, turning decomposition into a sustained disturbance rather than a brief pulse of nutrients. This contrast also complicates human responses. Removing sargassum indiscriminately from the ocean would damage habitat used by animals offshore. Allowing immense deposits to decay untouched beside populated or ecologically sensitive shores can create different harms. Management must therefore distinguish floating ecosystems worth protecting from coastal accumulations, requiring careful removal while avoiding nesting turtles, excessive sand loss, and damage from heavy equipment. Since the Great Atlantic Sargassum Belt became persistent after 2011, this balance has had to be reconsidered across the Caribbean and tropical Atlantic. The phenomenon has returned year after year, although its size and distribution vary substantially between seasons. Some years bring extraordinary biomass, others less, and currents determine which shores receive the greatest burden. Sargassum itself has not changed its nature. The surrounding circumstances have changed its role. In open water, moderate rafts create complexity where little structure exists. Along a coast overwhelmed by accumulated biomass, the same living material can reduce light, consume oxygen, obstruct movement, and alter habitat. Ecology often turns on such thresholds. A nursery can become a covering, shelter can become obstruction, and abundance can transform the consequences of a species without changing the species at all. For centuries, each new instrument brought the Sargasso into sharper focus without bringing it fully into view. Nets revealed larvae, chemical sensors recorded changes in seawater, satellites traced floating vegetation across entire basins, and genetic sequencing exposed organisms too small to see. Yet the accumulation of knowledge has produced an unusual result. The better this sea is measured, the more precisely its unanswered spaces can be described. Among them is an event central to the life of Atlantic eels. European and American eels converge upon this region to reproduce, as larval distributions and migration evidence strongly indicate, but the act itself has never been witnessed in the wild. Researchers have not recovered their eggs from the open sea. The depth, timing, courtship behavior, and exact environmental cues surrounding spawning therefore remain reconstructed from indirect evidence. An animal can cross an ocean toward reproduction, while the decisive hours of that journey remain outside human observation. Navigation carries a separate uncertainty. European eels, departing continental waters, travel roughly 3,700 miles toward a region their adult bodies have never previously occupied. Experiments show that eels can respond to magnetic conditions, supporting the possibility that Earth's geomagnetic field contributes to orientation. Currents, temperature, pressure, chemical cues, and inherited behavior may also participate. None has yet been demonstrated as the complete mechanism capable of explaining a successful ocean crossing. Far above those migrating animals, another puzzle has appeared on a much larger visible scale. The Great Atlantic Sargassum Belt emerged as a persistent phenomenon around 2011, but its origin cannot be reduced to one proven trigger. Nutrient inputs, river discharge, atmospheric deposition, upwelling, circulation changes, and favorable temperatures have all been investigated. Their relative importance changes across seasons and years, leaving the bloom as a product of interacting forces whose thresholds remain incompletely resolved. Greater uncertainty lies thousands of feet below. Beneath roughly 2.5 million square miles of open water stretches a deep environment known from comparatively sparse biological sampling. Repeated research along particular routes can reveal valuable patterns, but a transect is still a narrow line across an immense abyssal landscape. Species distributions, food webs, reproductive cycles, and localized communities may exist beyond the paths visited by research vessels. At microscopic scales, the unknown becomes even larger. In 2004, Craig Venter and collaborators used environmental genetic sequencing on Sargasso seawater and uncovered more than 1 million previously unknown genes. That discovery did not amount to a complete inventory. It showed instead how much biological information could occupy what appeared to be ordinary seawater. Many microbial genes still lack clearly understood functions, and many organisms resist cultivation under laboratory conditions. Their interactions form networks that genetic fragments alone cannot completely reconstruct. Even decades of continuous observation have limits. The Bermuda Atlantic Time Series study provides an exceptional record from one part of the Sargasso, but no fixed station can represent every current, depth, season, or biological event across such a vast moving system. Satellites broaden coverage at the surface, while ships and autonomous instruments descend beneath it, each method illuminating a different slice of the whole. Western navigators recorded the Sargasso as early as 1492, and systematic modern observations have since transformed drifting impressions into measurements of chemistry, circulation, biology, and climate. The remaining gaps are no longer merely blank areas on a map. They have names, evidence around their edges, and increasingly precise boundaries. That may be the most revealing result of studying this sea. Knowledge has not erased its mysteries. It has separated genuine uncertainty from speculation, showing exactly where observation ends. Within those boundaries, eels still reproduce unseen, microbes carry unfamiliar genes, deep communities remain sparsely sampled, and vast blooms arise from combinations science can describe without yet fully predicting. A boundary drawn around the Sargasso Sea would cross no coastline, meet no customs post, and belong to no single government. Its defining edges are currents rather than territory. Bermuda rises within the wider ecosystem, surrounded by waters under its jurisdiction, but beyond that zone, most of the Sargasso extends across the high seas, where authority is divided among international rules rather than concentrated in one coastal state. That legal geography creates a peculiar mismatch. The animals using this sea recognize no jurisdictional boundary. Eels arrive from rivers across Europe and North America. Turtles enter as young drifters and later depart toward distant feeding grounds. Migratory sharks, tunas, billfish, whales, and seabirds pass through waters governed by different agreements depending on the activity involved. A fishing vessel may answer to one international body, commercial shipping to another, while other pressures fall under separate conventions or national obligations tied to the vessel's flag. For much of modern history, there was no single institution empowered to manage the Sargasso as one connected ecosystem. Existing organizations could regulate particular sectors, yet none possessed comprehensive authority over the sea itself. Protection therefore resembled a mosaic whose pieces had been designed for different purposes. The ecological whole existed clearly in nature long before international governance had a framework capable of treating it as such. Bermuda helped push that problem into diplomatic view. On March 11, 2014, representatives gathered in Hamilton to sign the Hamilton Declaration on Collaboration for the Conservation of the Sargasso Sea. The original signatories were the governments of the Azores, Bermuda, Monaco, the United Kingdom, and the United States. Others later joined, including the British Virgin Islands, the Bahamas, Canada, the Cayman Islands, and the Dominican Republic. From that declaration came the Sargasso Sea Commission, built around scientific expertise and international cooperation. Its role is stewardship, keeping the health, productivity, and resilience of the ecosystem under review while encouraging governments and established organizations to act. Yet the declaration itself is not legally binding, and the Commission has no independent management authority. It cannot simply close a fishing ground, impose a fine on a passing ship, or create enforceable law across the high seas. The distinction matters because ecological processes continue regardless of whether governance is voluntary or compulsory. Plastic can cross national boundaries with currents. Migrating animals can spend one season under coastal jurisdiction and another beyond it. Carbon dioxide, absorbed from an atmosphere shared by every nation, alters seawater without regard for political maps. A species may therefore depend on protection across an entire migration while encountering very different legal conditions along the route. International law has now begun to change that landscape. In 2023, nations adopted the agreement under the United Nations Convention on the Law of the Sea, on the conservation and sustainable use of marine biological diversity of areas beyond national jurisdiction. After reaching the required number of ratifications, the agreement entered into force on January 17, 2026. For the first time, a global, legally binding framework specifically addresses biodiversity across areas beyond national jurisdiction. It provides mechanisms for creating area-based management tools, including marine protected areas, establishes procedures for environmental impact assessments, addresses marine genetic resources, and supports capacity building and technology transfer. The Sargasso now exists within a legal era that earlier generations of researchers did not have. Still, a treaty entering into force is different from an ecosystem becoming protected overnight. Governments must participate, institutions must function, proposals must be developed, scientific evidence must guide boundaries, and agreed measures must be implemented. The currents enclosing the Sargasso have always made it one ecological place. Human law is only beginning to acquire the tools to recognize that same unity. The Sargasso Sea becomes easiest to misunderstand when it is treated as an isolated patch of Atlantic water. Its surface may appear geographically remote, yet the forces passing around and through it belong to systems that reach continents, fisheries, rivers, atmosphere, and the lives of animals separated by thousands of miles. Isolation here is only partial. Beneath it lies connection. Along the western edge, the Gulf Stream carries immense volumes of warm water northward before continuing into the broader North Atlantic circulation. That transport contributes to the comparatively mild climate of northwestern Europe, although winds and atmospheric circulation also play major roles. The Sargasso occupies the interior of this moving architecture. Changes around its boundaries therefore belong to a larger ocean system whose influence extends far beyond the floating sargassum. Biology makes those connections even more intricate. European and American eels converge upon this region to reproduce after lives spent across separate continents and watersheds. Loggerhead turtles use floating vegetation during vulnerable early stages after leaving nesting beaches. Young tuna, billfish, mahi-mahi, and many smaller fishes find food and structure among the drifting growth. Microorganisms invisible from the deck transform nutrients and carbon, participating in chemical cycles that continue through the Atlantic and atmosphere. None of these relationships operates alone. A current can transport heat, larvae, dissolved gases, nutrients, and human debris at the same time. A floating habitat can shelter an animal while carrying synthetic fragments beside it. A microscopic cell can influence carbon chemistry while responding to temperature and nutrient availability. The Sargasso gathers these processes into one region, revealing how difficult it is to separate physical oceanography from biology, chemistry, climate, and human activity. Its greatest scientific lesson may therefore come from the gaps between measurements. Satellites can repeatedly observe the surface, but cannot identify every organism below it. Research vessels can lower instruments through thousands of feet of water, yet each cast samples only a narrow column within an enormous moving sea. Genetic surveys can reveal unfamiliar sequences without immediately revealing what the organisms carrying those genes do. Tracking devices can follow an eel across much of the Atlantic and still fail to witness the final act of reproduction. Scale complicates knowledge further. Some changes unfold within hours as plankton respond to sunlight. Others emerge across seasons, decades, or generations. Deep water preserves signals that began far from the place where scientists eventually measure them. Animals respond to cues humans may barely detect, including gradients of temperature, chemistry, current, odor, and Earth's magnetic field. To understand a migration, researchers must reconstruct not merely where an animal travels, but what information its nervous system extracts from an ocean that is itself changing. That difficulty has become more consequential as familiar environmental cues shift. An eel's inherited navigation evolved under conditions assembled over immense spans of evolutionary time. Today's migrants encounter altered temperatures, changing acidity, artificial contaminants, modified food webs, and coastlines transformed along both ends of their journey. Scientists can measure many of those changes individually. Determining how they interact inside a migrating animal remains much harder. This is where the Sargasso resists becoming a completed story. More than 5 centuries after European sailors recorded its floating weed, and generations after systematic oceanography began probing its interior, observation continues to expose additional layers rather than a final explanation. Aristotle could examine eels and wonder where they came from. Modern researchers carry satellites, genetic sequencing, autonomous instruments, and decades of measurements, yet the animals still disappear into open water, carrying part of the answer with them. The Sargasso does not diminish science by keeping those answers incomplete. It shows the scale of the task. Across a connected ocean, knowledge advances through fragments gathered patiently, while the living system continues moving between them. There is no single moment when an ecosystem becomes different from the one inherited by the generations before it. Change accumulates through missing animals, altered journeys, unfamiliar chemistry, and pressures that overlap until an older pattern becomes harder to find. In the Sargasso Sea, some of those absences can now be measured, while others remain hidden inside a history that science never recorded in full. The European eel carries perhaps the clearest numerical warning. Recruitment of young European eels has fallen by more than 90% compared with levels observed before the major decline that became evident in the 1980s. The International Union for Conservation of Nature classifies the species as critically endangered. No single cause explains that collapse. Dams and other river barriers interrupt migrations. Wetlands have disappeared. Fisheries have removed eels at several life stages. Parasites, pollutants, changing currents, and a warming climate add further strain across a life cycle stretching between fresh water and the open Atlantic. Each pressure occurs somewhere different, yet all act upon the same animal. An eel surviving contaminated rivers must still cross coastal waters, navigate thousands of miles, reach its spawning region, reproduce successfully, and leave offspring capable of returning toward distant continents. Failure at any stage reduces the number completing the cycle. Conditions within the Sargasso may therefore matter alongside damage occurring far beyond it, even though the relative influence of each threat remains difficult to separate. Sea turtles carry another version of that vulnerability. Loggerheads use floating sargassum during their early oceanic years and are considered vulnerable globally by the International Union for Conservation of Nature. Hawksbill turtles, also associated with floating sargassum habitat, are critically endangered. Their decline began through pressures extending well beyond this sea, including harvest, accidental capture, coastal development, and degradation of feeding and nesting habitat. Yet young turtles entering the Atlantic today also encounter synthetic debris woven through the same drifting structures that provide food and shelter. Smaller residents receive less attention because many never leave the floating habitat at all. Sargassum fish, pipefish, shrimps, crabs, snails, and other invertebrates inhabit a world measured in branching fronds and pockets of water. For them, changes to the weed are changes to nearly everything around them. Plastic fragments can become part of that structure. While temperature and chemistry alter conditions at scales, these animals cannot escape through continental migration. Calling the Sargasso a dying sea would flatten this complexity. Life remains abundant. Sargassum still drifts. Larvae still develop. Microorganisms still transform nutrients, and migratory animals continue to arrive. What has changed is the background against which those ancient relationships operate. Evolution shaped them under ranges of temperature, chemistry, food availability, and seasonal timing that are now shifting within only a few human generations. The deepest uncertainty lies in the missing baseline. Systematic measurements began after industrial fishing. Widespread chemical pollution and much coastal transformation were already underway. Plastic entered the ocean before researchers possessed today's monitoring networks. By the time long records became possible, the Sargasso had already experienced decades of human influence. Even now, enormous portions remain biologically unobserved. No camera has recorded wild eels spawning. Vast reaches of the abyssal plain have never been comprehensively cataloged. Microbial surveys reveal genetic diversity faster than researchers can assign ecological roles to it. Species may decline before their relationships are understood, and communities may shift before anyone has described their earlier state. That leaves the Sargasso carrying two histories at once: the measurable record of recent change, and an older record preserved only imperfectly in organisms, sediments, currents, and evolutionary inheritance. Science can reconstruct parts of what came before, but never recover every missing detail. The ocean was already ancient when systematic observation began, and much of what remains known today survives without a complete account of what has already passed away. A coastline gives the eye somewhere to stop. Sand, rock, harbor, cliff, or marsh can mark the place where ocean becomes land and where a name can be attached to a shape. The Sargasso Sea offers no such ending. Travel outward from its center, and there is no beach announcing departure, only water changing gradually as currents loosen their enclosure and the Atlantic becomes another part of itself. That absence of shore has followed the Sargasso through human imagination. Ancient Mediterranean sailors may have encountered floating weed in the Atlantic, and medieval cartography recorded the region before the voyages that made it famous in Europe. When Christopher Columbus crossed these waters in 1492, his crews watched vegetation gather around their ships far from land. Later centuries supplied darker legends, transforming drifting weed and becalmed vessels into stories of entrapment. Science eventually replaced much of that mythology with measurements, yet the sea proved stranger in ways the legends never anticipated. Names accumulated because no single description could contain it. Oceanographers recognized a nutrient-poor subtropical gyre. Biologists found floating habitat crowded with specialized life. Geneticists encountered microbial diversity hidden inside a cup of seawater. Long-term monitoring turned the region into a record of changing ocean chemistry. Each description captures one layer, while thousands of feet beneath the familiar surface, other layers remain scarcely observed. Near the deepest parts of the region, the seafloor lies roughly 14,800 feet below the sunlit surface. Human beings have sampled portions of that depth, but vast areas remain biologically unexplored. No complete inventory exists for the animals moving across the abyssal plain. Higher above, another mystery continues its migration. Adult eels disappear into the Sargasso after journeys from distant rivers, yet their spawning behavior in the wild has never been directly witnessed. Their transparent larvae eventually begin moving outward, carried into current systems leading toward continental waters they have never encountered. Even the floating sargassum contains a history that cannot be reconstructed perfectly. Researchers can measure plastic among its branches today, examine archived specimens, and compare modern observations with earlier records. They cannot return to an untouched Atlantic and measure exactly how these communities functioned before industrial contamination altered the background. Part of the original baseline disappeared before instruments were present to preserve it. This incompleteness belongs to a much larger ocean. Water covers roughly 71% of Earth's surface, and more than 80% of the ocean remains unmapped, unobserved, or unexplored in detail. The Sargasso is remarkable precisely because it has received sustained scientific attention. Yet decades of ships, satellites, instruments, genetic surveys, and drifting sensors have not exhausted what can be learned there. Greater observation has enlarged the catalog of unanswered things alongside the known ones. Somewhere beneath the moving surface, organisms rise and descend according to rhythms older than recorded history. Far above them, sargassum gathers into temporary neighborhoods, separates, and gathers again. Eels arrive carrying biological instructions inherited across generations. Their larvae leave without memory of the rivers toward which currents will eventually carry them. Around all of this, the gyre continues its broad circulation without requiring a coastline to give it form. Human maps need boundaries because boundaries make places manageable. The Sargasso exists by another geometry, shaped by motion, temperature, wind, and current. Its edges breathe across the Atlantic, never fixed in precisely the same place. And in water, thousands of feet beneath any passing ship, a small living body may produce a brief pulse of its own light. It needs no witness. The flash appears, fades, and becomes part of an ocean whose oldest stories were unfolding long before anyone learned to name the sea above it.